This is lecture 17, Principles of Metabolism, and in this lecture we will look at some properties of experimental systems, things we have to consider when measuring metabolism. So there are many ways to study metabolism, and as often in biomedicine we have this trade-off that you can either be very close to actual human physiology, but then you typically have problems doing very precise measurements because there is, of course, limitations to what you can do with humans. And on the other hand, there are model systems, cells or animals and stuff, where you can do very precise measurements and very precise experiments. So you have high precision, but then they tend to be far away from the physiological situation you're actually interested in. So we tend to have this type of trade-off, and we would like to be over here where everything is fantastically well measured and perfectly captures human physiology, but there's hardly anything like that. So instead we tend to have this type of trade-off. So there are very specialized in vitro systems, cultured cells, or even proteins in vitro pathways just studied in lysates, where you can do very sophisticated technical things, but they are far away from physiology. And then there is this whole sort of a gradient from this towards actual human studies. And on the way, we can think about other things that you can culture, organelles, tissue sections. You might be able to perfuse some organs, so not in vivo, but ex vivo. Or you can think about going to in vivo studies or do physiology measurements or measure metabolites in plasma. But all of these approaches have their pros and cons. So we'll look at a couple of these. So we'll start with the most kind of artificial system, but one where you can do very precise measurements and which we work with a lot in our lab. And that's, of course, cell culture, batch cultures, so cells in flasks or in dishes in incubators. And this tries to recapitulate the most sort of basic properties of physiology. You have the right temperature, humidity, CO2 to capture the pH, buffering and so on. But of course, it is far away from cells in the body. There's major problems with attachment to substrate. Cells differentiate in these conditions and are no longer like the ones that we have in the body. So there are problems with the cell system itself. And there are also some issues specific to metabolism, trying to keep a reasonable nutrient level and things like that. We'll look at this a bit in detail. So this is problems with the culture system, but on the other hand, we can do sophisticated measurements. We can do uptake-release measurements. Isotope tracing is fairly easy, and so on. So there are other things we can culture though, so we don't have to restrict ourselves to cells. Also in the same batch culture type of setting, people culture organoids, this type of somehow differentiated little structures that you can grow out of certain cell types. So that gives you a bit of the structure that's available in vivo, and you still have the same type of measurement abilities that you have in cell culture. There are also some people who culture tissue slices, and this is an interesting kind of intermediate model where you have the intact tissue separated out from an animal or even from patients if it's something that is resected and discarded. And so this can be sliced up while cells are living, and they can be cultured that way. So there are some options already with culture systems. So let's look at a couple of specific points with cell culture that can be a bit problematic. So oxygen is one important point. So you may have seen that some cell types, stem cells particularly, can be difficult to culture in atmospheric oxygen tension. So people need low oxygen incubators to be able to grow those cells properly. So they really don't tolerate oxygen. So there's a double-edged sword with oxygen. If you have too little oxygen, it will of course hinder respiration. So you will have very glycolytic cells, or they might not survive at all. There are also some oxidases in some metabolic pathways that are dependent on oxygen themselves, so they could be blocked. Cholesterol synthesis, for example, requires oxygen. There's an oxidase step that needs that. So that can be blocked. But if you have too high oxygen, you can start oxidizing some molecules that are important for cells. So this is usually called oxidative stress. Tetrahydrofolate is one example. This is an important carrier from methyl groups, and it has these reduced hydrogen groups that get attacked by oxygen. And this one can start oxidizing and not function well if you have too high O2. Thiol groups is well known, so cysteine groups, SH groups, they oxidize easily and you get disulfides, and this can also inactivate proteins and enzymes. So high oxygen can be a problem for some cells. But so where should oxygen be? What oxygen should we have if we want to culture a particular cell? So what is norm oxia? What's the normal level? Well, this is not so easy to say because even in vivo it varies a lot by tissues. So there are some very low oxygen tissues in some parts of the body. But if you think about epithelium in the lung, it's probably at pretty high oxygen concentration. So it varies a lot, and I think in many cases we don't know what the appropriate oxygen concentration should be for a particular cell type. So that's a problem, a fundamental problem, I think. And there's also the situation that the actual oxygen that cells experience is not what you have in the atmosphere above your cell culture. It's what oxygen you actually have at the bottom. And when people measure actual oxygen tension in the medium in a cell culture flask, it declines quite drastically sometimes from the sort of surface of the medium down to the bottom. So in many cases, we actually don't know what oxygen concentration our cells are experiencing. Maybe it's actually quite low. So this field is, I think, in its infancy still. We don't know exactly what oxygen tensions we have or we should have. But this is clearly a problem. A few points about medium, cell culture medium, and batch culture effects. So there's a lot of different cell culture media around, different formulations. A lot of people use minimal medium, DMEM, RPMI, those are common things, but there are several others. And if you start looking at the concentrations of nutrients in these, they vary quite a bit sometimes. So glucose, for example, is famously high in DMEM. It's 25 millimolar, 5 times physiological, twice as much as physiological in RPMI, around 10. Eagle's original medium, one of the first formulated, was physiological in this sense. It was 5 millimolar. And you can see there are some amino acids which are present in some medium but absent in others. And some of them just vary a lot in concentration. Twofold variations in some amino acids is not uncommon at all. Glutamine tends to be very high in medium compared to physiological situations, but this also varies quite a lot. And then there are some particular compounds that have been added in some media and not in others. RPMI, most formulations contain hydroxyproline. It's not entirely clear why, but it does. Somebody felt that this was important. And this is because these culture media have been developed kind of in brute force sort of fashions, optimized to have as much cell growth as possible. And usually for some particular cell type that the investigator was interested in. So it's optimized for that, and they picked whatever concentrations seem to work to get their cells to grow as fast as possible. And it's not always designed to be physiological. That was never the purpose. So this is a problem, and I think culture media has been in some ways developed for other purposes, for using cells as model systems, for molecular biology or for something else, studies a particular pathway in a cell. And then you just want to have as much cells as possible growing and being healthy, and so it's easy to do culture. But maybe we should change this and we should go to more physiological medium, even if it means that the cells will grow twice as slowly. So this is something that has to be reevaluated, I think. Some media also have vitamin deficiencies. So DMEM, for example, doesn't have cobalamin, vitamin B12. And this can cause some cells to change, and they can lose some enzymatic activities which are dependent on these vitamins. So that can change 1-carbon metabolism and methylation, for example, if you lose B12. So that's also some not so easy to spot sometimes, these differences, but they can have effects on metabolism. So medium formulations matter. If you look at how metabolism is affected by nutrient concentrations, it's quite interesting. So glucose, obviously, with these large differences can cause large differences in metabolism. So this is just some unpublished data. We just looked at behavior of some cells in different glucose concentrations. And for example, this cell line A498, you can grow this very well in either 2 millimolar glucose, so below physiological levels, quite low, or 10 millimolar, twice as high as blood glucose. And in either case, they grow just equally fine. But if you look at glycolysis and glucose uptake, it pretty much scales with how much glucose you give them. So you will think that the cell is very glycolytic if you give it a lot of glucose. If you give it less, you will think that it's more oxidative. So this is clearly an issue, and something that hasn't been evaluated much. And then with batch culture specifically, there is the obvious problem that you have to do medium changes all the time because your nutrients continually get depleted by the cells and waste products tend to build up. And this is one reason we have usually higher concentrations of nutrients in medium than in blood, because we kind of need to give a bit of a store and sort of buffer it up a bit, because if a physiological concentration is here, we want to start a bit above it so that we don't get too far below it. And this is also a fundamental problem with batch culture. It's kind of unavoidable. Then there are a few biochemical artifacts that are good to be aware of. So glutamine is a problem, for example, in cell culture or anywhere you give nutrients to cells, because glutamine degrades in water. It's one of the amino acids that's actually fairly unstable in water. So what happens is that this particular amine group is a bit reactive, and this can sort of flip back and cyclize, and you get sort of a form of proline, oxoproline, is generated by this reaction, and you lose an amine group. And so this happens spontaneously, and it has about a half-life of 1 week. So glutamine degrades fairly quickly in culture conditions. So these things happen, and there's also the possibility that you can have enzymes in your serum that can attack other nutrients or sort of change your measurements, because they actually convert your nutrients in your metabolites into other things. So one important determinant here is to heat inactivate the serum, so that can kill some of these enzymes and sort of relieve that problem. But that's something to be aware of. And for measurements, it's also the case that there are some compounds that are volatile just by nature, and they can evaporate during cell culture. And so if you are looking for these, you might not find them because they might simply go away while the cells are being cultured. Putrescine is a polyamine, and is an example of this volatile compound. So can we prove upon this? Well, there are some continuous cell culture systems. They definitely don't get rid of all the problems with medium formulations, but at least you get rid of the batch culture problems. So you can have a more stable environment, somehow simulate the circulation so that you get fresh medium in, sort of like getting fresh blood in, and then you have a way of getting rid of the waste. So there are these contraptions. People build this Nutrostats. This is a recent example from a Nature paper from David Sabatini's group. And sure enough, in these systems, you can ensure that you have a robust and physiological glucose concentration, for example. So that's one possible improvement you can do to have cell culture be a bit more close to physiology. The next step would be to do some type of perfusion of an actual organ. That's, of course, a way of getting at a more physiological situation. So these are fairly difficult things to do, but there are groups who do it, where you can take, for example, a liver, sort of isolate it, and you can perfuse it in systems. Again, trying to sort of simulate a blood circulation and keep the tissue alive and in good condition. And you can set up these so that you have ways of getting at samples. So you could introduce, for example, isotopes in these systems and look at tracing and wait until a system like this settles into steady state, or whatever you want to do. Take out metabolite abundance measures and things like that. So these systems is one more physiological alternative. And then we have the human plasma measurements. So this is for sure a physiologically relevant type of data. It reflects human physiology, but it has also some issues with interpretations that I think we should be aware of. So one thing with plasma data in general is that it is kind of a reservoir that is affected by a lot of different organs that could have different metabolic profiles. So you can think of it as kind of integrating whatever effect different organs have. And so this means that what you will see might not be so easy to interpret, because a change in the metabolite could originate from various different organs. Or it could mean that if you have a smaller metabolic change in some particular organ, it might sort of drown out in the noise. It might be averaged out and you won't see it. So that's a bit of a problem. Another issue with plasma measurements, which is not talked about so much perhaps, but I think it is important, is that this is a very particular type of blood. This is venous blood draining the muscle in the forearm. Put the needle in a vein, obviously. Arteries are not so easy to cannulate. And that's a very particular type of blood. So I think that it's interesting and important to think about what nutrients are actually presented to tissues. And what we get from the diet is normally extracted from the gut. All the nutrients, it goes into the portal vein and ends up in the liver, and the liver filters this, and then it goes to the major circulation and into arterial blood. And then that is presented to most peripheral tissues. So that would be the type of blood we would like to look at, I think, in many situations, if we want to study the nutrients given to a particular tissue. But that's of course not what we see. When we look at venous blood from this typical blood drawing session from a human, we see the blood after being filtered from over muscle. And of course the muscle can take up some of the nutrients, clear some stuff, release some amino acids, and whatnot. So that's something to be aware of. Along the same lines, lymph is also a component that's important to think about and often forgotten. So this drains into the vena cava, so again goes into the major circulation. And again, components of lymph will be presented to a lot of tissues, but when we look at venous blood, we will see this filtered by muscle. And pretty much all data available out there, all the tons of data we have in human plasma is from this situation. So we should be aware that this could be a bit of a distorted picture. And then finally, sort of in between the human plasma sampling type of data and cell culture studies, there are some experimental systems where you can look at either humans or more commonly animal models and look at particular veins or arteries in the body or particular tissues and do more precise measurements. But this is, of course, very difficult to do, particularly in people. So there are few of these studies and far between. But there are some, and I think that's very valuable data and very interesting to look at. So to the left here is shown this graph. We've looked at this before in the amino acid lecture. This is from a review paper by Philip Fellig. The data was done by him and by John Varén here at Karolinska. So they did actual cannulation of healthy volunteers and looked at the arteries and veins that feed and drain muscle and that feed and drain the splanchnic bed, so gut and the liver. And so this is difficult studies to do. You have to cannulate people, and case of the liver, you have to access the portal vein, and that can only be done in surgery, so this was not easy to do. But they managed to do it, and the data is really valuable. So here they sort of defined the type of amino acid exchanges that actually occur in these organs. That really helped understand their metabolic physiology, their mode of metabolism. So this is valuable, but hard to do. In animal models, of course, you can do more things. There are plenty of examples of people looking at particular tissue beds or arteries in animal models. And this is an important source of data, I think. This particular picture is from a tumor model, where it's an implanted tumor in vivo in rats. And this is done in a way so that the tumor is fed and drained by particular veins and arteries. So you can cannulate those, and you can look at what is the tumor taking up or releasing. And then, of course, you can introduce tracers if you want into these systems, and you can take out material from the tumor after being fed by tracers. And so you can do all sorts of type of metabolic studies in these systems. So it's not easy, requires a bit of surgical expertise and so on, but I think these are really important systems that we should see more of.